begin. Hi, okay, welcome to week 2, lesson 2. And I'm going to be putting this in 2 presentations because there's so much info in this week's lesson. So tonight I'm going to be talking up to about slide 15. I'll be covering and finishing up with cell wall, bacterial cell walls. And then the second presentation, I'm going to be talking or picking up with slide 16 until the end. Okay, so anyhow, the first question is, it's been stumping people for years, is what is life? After all, microbiology is life. And what happens if I told you they have not universally accepted any definition, one definition of what is life? What we do know is that all living things have 4 basic attributes. And one is that they should be able to use matter. A second one is that they can maintain homeostasis. So what we mean by homeostasis is that something living can internally, physically, and chemically keep things in balance. They can keep things in balance. The third attribute is that a living organism can evolve and adapt. And finally, the fourth is that it can reproduce. Now, where the argument lies of what is life is that some biologists say that viruses are living because they can adapt and evolve. And if we look at even the COVID virus, that right now it has evolved several variants. So it can adapt and evolve. And also, viruses do have the ability to reproduce. It is subjective though. They reproduce within our cells. So there is the final judgment on this about viruses is still out. Okay. On the next slide, what we do agree in, or what is established as theory, is the cell theory. And this came about by the work of 3 gentlemen or scientists, Theodor Schwann, Matthias Schleiden, and then Rudolf Virchow. So what has been determined is that cells are the smallest basic unit of life because they can use matter, they can maintain homeostasis, they can evolve and adapt, and they can also reproduce. All living organisms are composed of 1 or more cells. Again, the cell is the basic smallest unit of life, and that cells arise from pre-existing cells. So these three principles are held as truth. So if cells are the basic unit of life, what must they have? At a minimum, all cells must have these 4 things. Okay, they must have a cell or plasma membrane. They must have DNA and RNA, ribosomes, and enzymes. These enzymes are needed for the chemical reactions that will break down or use this matter. There are 2 main types of cells in prokaryotes and eukaryotes. Comparing the 2, one thing that you may note is that eukaryotic cells are much larger and have more stuff than the prokaryotic cell. The eukaryotic cell has cell organelles, which the bacterial cell is lacking. They do have the ribosomes though, and they do have a cell membrane, DNA and RNA, and enzymes. So I'm going to be talking right now about some of the structures of the prokaryotic cells, getting into depth a little more with that. And then at points I'll be comparing both prokaryotic and the eukaryotic cells with the structures that they have in common. So the characteristic of prokaryotic cells, first of all, we know they are usually exist only as one cell, and they are microscopic. What they do have is their genome, which are all their genes, are organized in a nucleoid. And I'm going to explain that in the next slide, but they have no nuclear membrane around their genome, which eukaryotic cells have. We've got that nuclear membrane, also aka known as nucleus. But the bacterial cells do have DNA. Okay? And then again, the two major groups of prokaryotic organisms are your bacteria and your archaea. Bacteria we're going to be working with in lab, especially with your skin project bacteria throughout the semester. Archaea are prokaryotes, but again, we won't be working with them because first, we cannot grow them in the lab. Why? Because they are extremists. Many of them survive in very extreme conditions, hot temperatures, very cold temperatures, high salty environments. And they are built that way to survive under those conditions, so we can't grow them in the lab. One thing, though, that's kind of interesting about the archaea is that they have not been found to cause disease in humans. So they are a friendly group, very friendly group. But they just live on the edge. They are very extremist. Okay. Now, genes are basically located on a chromosome. And the nucleoid is basically a chromosome containing these genes. And these genes on the chromosome are located in the middle or in the interior of the bacterium, and this is known as a nucleoid. What is anchoring these this chromosome into a specific location within the bacterial cell are these special DNA-associated proteins. They serve as anchors. So if you look at that top picture there, you have a bacterial cell with the electron micrograph, and where you see the green dashes, that is showing you where the nucleoid is or located within the bacterial cell. We do have some, it looks like those little letter M's. Those are your DNA-associated proteins that kind of keep that chromosome anchored or formed within the center of the bacterial cell. There is no nuclear membrane containing that chromosome. One thing about the chromosomes of bacteria, they are circular. Our cells, the DNA, the chromosomes are linear, okay? So usually bacteria cells will have one, maybe two chromosomes, and they are circular. Now, some bacterial cells, not all, but some may carry extra genes on plasmids. So if you notice in the slide here, they are pointing out the plasmids, the little smaller rings within the bacterial cell. And these plasmids do carry additional genes. The genes are not essential for the bacterial survival, but they do come in helping, maybe providing antibiotic resistance, fertility, and we will get into that later on too in the semester. So again, bacterial cells have a nucleoid, and they may also contain plasmids. Now here I'm talking about the ribosome structure, and I'm going to compare the prokaryotic ribosome structure to the eukaryotic ribosomes. What they both have in common is that the ribosomes, first of all, contain rRNA and some proteins. The function is the same. We use the ribosomes, or the ribosomes are used for protein synthesis. Another thing is the ribosome structure is also made up of two parts, or two subunits. A larger subunit and a smaller subunit. So if you see here, we've got the two subunits here for both eukaryotic and prokaryotic cells. And those two subunits, when it comes to making a protein, will then kind of connect and form your complete ribosome. Now, what is different with the prokaryotics and the eukaryotic cells is the smaller subunit, this guy right here, is made up of different RNA. Okay? Different RNA. And it shows on the slide here the green circled 16S for the prokaryotic cells. That's what makes up, that's the gene that makes up the smaller subunit for prokaryotic ribosomes. Down below the eukaryotic ribosomes, the smaller subunit is made up with 18S gene. So the gene sequencing is different between prokaryotic ribosomes and eukaryotic ribosomes. What's important with this is, guess what? We use this sequence to analyze and help us identify the bacteria and also its relationship among other bacteria. These sequences, 16S, yes, all bacteria have it, but it may be just a little different. A little bit different in the sequence of the nucleotides. And that will depend upon the type of bacteria. So we use this guy right here in DNA sequencing for the identity and also the relationship of the bacteria among other species of bacteria. Very important tool that we use now because we've got the technology. So that is the similarity and the differences between both prokaryotic and eukaryotic ribosomes. The next slide here, cell membrane. Okay, we do have cell membranes in both prokaryotic and eukaryotic cells. And the cell membrane is made up of a phospholipid bilayer. Okay, 2 layers of phospholipids. And they are similar in both types of cells, except the prokaryotic cell is lacking in sterols. That's what the eukaryotic cells, which is example of that would be cholesterol. Okay. The cell wall bacteria have is in about 90% of your bacteria will have a cell wall. And they have a very important function. They help maintain the cell shape. They also resist invasion, kind of like a gated community. They will help in protecting the cell from invasion of other pathogens or other external substances. The cell wall may help in invading a host. And also the cell wall may have properties that help or aid in causing disease. So now I'm going to talk about the structure of the cell walls, and especially the component found in many bacteria that make up the cell wall. And that is called peptidoglycan. So if you look at the word peptidoglycan, you'll see, let's break that down. The first part of the word is peptide, P-E-P-T-I-D, peptid. Okay, peptid sounds a lot like peptides, okay? And peptides are amino acids. The second part of the word, G-L-Y-C, looks like glucose, or sounds a little like glucose, sugar. So take that word apart, and that tells you what peptidoglycan is made up of. Sugars, or derivatives of sugars, and peptides, amino acids. So the sugars, let's talk about them. They're the main guys. The sugars are NAM, and NAG, NAG. And NAM will stand for N-acetylmuramic acid, and your NAG is your N-acetylglucosamine. So these are two sugar derivatives, and what they do is they pretty much connect by covalent bonds, alternating into a long chain. So you will have a NAM and then a NAG, a NAM and a NAG, and they will form long chains. They are interconnected. The rows and the layers are interconnected with peptides or the amino acids and peptide short segments of peptides, 4 or 5 amino acids. So what does this form? This forms a strong yet flexible covering outside of the cell membrane, the cell wall. Now there are 3 types of bacterial cell walls that contain this peptidoglycan, and you will be dreaming about these because we will be talking about them and performing these stains and identifying bacteria based on this. Even in the hospital, we still routinely do these stains to determine whether gram-positive, gram-negative. And finally, the acid fast. We don't perform that in the lab anymore, but the gram-positive and negative stains we do perform, and it determines their cell wall, which is important in diagnosing and treating a disease. Okay? So the first one, the gram-positive, way on your left there, on the left-hand side of this slide. We're going to be now working from the cell membrane out into the cell wall. So at the bottom of that, we've got your nice little cell membrane. And then if you see in the center part, we've got layers of peptidoglycan. Okay. Layers of chains of peptidoglycan. You will have this peptidoglycan anchored into the cell membrane by teichoic acids. So those are the important structures you need to know with the gram-positives. They have layers, many layers, depending upon the gram-positive bacteria of peptidoglycan, anchored into the cell membrane by teichoic acid. There also is a simple layer of proteins, the S layer. That makes up your gram-positives. The second group, the gram-negatives, are a little different. They do have the cell membrane. They have a big empty space here called periplasm. But if you notice that the peptidoglycan, my gosh, one layer, one row, and that's it. Gram-negatives do not have a lot of peptidoglycan. And this will affect the way they stain. What they do have, though, is they have lipoproteins, not teichoic acid, but lipoproteins that connect to an outer membrane. Connects to an outer membrane. And this outer membrane has phospholipids, but it also has an additional structure embedded in that outer membrane, LPS, which stands for lipopolysaccharide. So these are just specific to your gram-negatives. And what's important with these lipopolysaccharides is that they contain lipid A, and lipid A is a toxin. So the toxin for this type of bacteria, your gram-negatives, is contained in the lipid A part of the lipopolysaccharide found in the outer membrane. Okay. And finally, we've got the acid fast, which my head is blocking the name acid fast, but it is there. These guys do have peptidoglycan, and we go way down here, right outside again of the cell membrane. But they have additional stuff, arabinane and galactan, which again are a type of sugar, lying right outside the peptidoglycan. And the important part, structure or molecule, are your mycolic acids. So again, if we look at this down here, if I can get the arrow there, we've got your peptidoglycan, a few layers. We've got your galactan and arabinane, and then we've got these mycolic acids. Mycolic acids just lying beyond the arabinane, connected to the arabinane. Those mycolic acids are waxy, so they're kind of like wax. It doesn't allow for regular gram staining. We have to do a special stain, acid fast. So there are a few bacteria that have this kind of a makeup in their cell walls. They all have peptidoglycan, but each one has special stuff. And I would ask that you make note of the special features for each one of these, okay? Okay. So now, what the peptidoglycan will do is it covers the entire bacterial cell. This covering is known as a sacculus. And again, I told you some of the features, or some of the important features, or things that these peptidoglycan can allow. And one of the attributes of the peptidoglycan is it contributes to the cell shape of the bacteria. So these now are some of the common cell shapes found in bacteria, and they can be either round, rod-shaped, curved, short rod, spirally, or even really tightly helical spiral. And they each have names, okay? So your round ones will be called coccus for one, or cocci. Your rod-shaped are called bacillus or bacilli. Vibrio are curved. They kind of look like a comma. Coccobacilli are going to be a combination of your round shape and your rod shape, kind of oval shaped. Spirillum are rather spirally. And then finally, the spirochetes are really a kinky spiral, really tightly wound. So in the last column here of the shapes, they give you the electron micrographs of how they look under the microscope. In the lab, we will mostly be seeing your cocci and also bacilli. Now, not only do they come in certain shapes, they also grow in arrangements. And they're named according to their arrangements, their shape and their arrangements. So, if we look at the second picture, they can be in pairs. And the name for that is diplococci. You've got two of them. Tetrad, they will grow in an arrangement of four, which is known as a tetrad. You may find a long chain of them, strepto, or string of them, and that will be in your picture here. Right here. String of cocci, string of round bacteria, known as strepto string cocci. Okay. Staphylococci would be a cluster of them. So it's not a real determined arrangement. It's just like a group of them. Sometimes when you see them under the microscope, it looks like clusters of grapes. That's known as staphyla, cluster. And then cauci is that they're round shaped. Now bacilli can be single, or you may also see them in chains, right down here. And in this case, we use the prefix strepto and bacilli because they are your rod-shaped. So you have a string of rod-shaped bacteria known as streptobacilli. As we work in the lab, you will become more familiar with these shapes and these arrangements. So practice makes perfect, but check out these names and these arrangements, just kind of going over them a little bit before we get to lab will help you. You won't feel quite as lost once we start viewing these. Okay. Finally, there are bacterial cells without peptidoglycan. So again, like I said, 90% do have peptidoglycan in their cell walls, but then we've got those, those little radical groups that just don't go along with the rules, and these bacteria are known or come from the class of mollicutes, and two genus or generas are your mycoplasma and your ureaplasma. So they don't have peptidoglycan. They aren't, and as a result, they're kind of pliable. They don't define or are not found in a specific shape. And if we look at them down here in this picture here, you're going to see, my God, some of them have tails, some of them are flat, stretched out, and some are even round. So they lack your peptidoglycan for structure, but this makes them pliable. So these bacteria can live deep within tissues, and I think that's what serves them. Without having the peptidoglycan, they are able to get there and stay there because of their lack of it. Okay, other cell walls that lack peptidoglycan are in your group or domain of the archaea. They lack peptidoglycan. What makes them different than the mollicutes is that their cytoplasmic membrane is different, and also their structure. The structure of the membrane is different. And instead of peptidoglycan, they may have other things or nothing at all. So what makes their membrane different is that it's not a phospholipid bilayer. It is a bilayer, but basically the tails of the phospholipids are made of a different molecule than what is found in eukaryotic or other prokaryotic organisms. And in this case, they're comparing bacteria phospholipid to the archaeal phospholipids. And as you see, the fatty acid tails are different. It's made of a different component. And even the heads are different too, made up of different types of molecules. And we have two examples here, A and B, of the archaeal cell boundaries. We have on the top there the membrane, but it is lacking peptidoglycan altogether. And also in the B picture, it does have some type of a component in the space there, but it's pseudomerium. So it's not the same structure as peptidoglycan. Okay. And again, these features of your archaeal cell boundaries help it survive extreme environments. Okay, I am going to end there. for this lecture, but I do want to tell you that there is an animation that you can view if you wish on the peptidoglycan, and the cell walls of your gram-positive and gram-negative is only last maybe about a minute or two. Give it a view, because sometimes animations help, at least help me in learning too. And then I will continue or pick up at this in the next lecture. Okay? See you soon.